my name's Tony Kolessa. I'm a graduate student with the MIT Department of Biological Engineering, where I work with Professor Paul Blaney and another graduate student, Jared Kay, on a technology that we think can contribute to solving a problem that lies at the center of modern medicine, that being antibiotic resistance. So antibiotics are one of the greatest advancements of modern medicine. I think that's illustrated by just the ubiquity of their usage. Every year in the United States, 250 million antibiotic prescriptions get written every year, and over half of US hospital patients actually receive antibiotics over the course of their stay. And this is because we're not only using antibiotics to treat infections which previously could be life-threatening and now we're turning them into just minor nuisances, but we're also using them prophylactically for reducing the risk of infection during surgery, as well as when we treat patients for cancer and other diseases where the treatment could actually ablate their immune system. But this fundamental part of our medical practice is actually at risk. So far, just in the past 20 years or so, we've now been observing resistance to all clinically deployed antibiotics. And in fact, this has progressed to the point where 1 in 4 of healthcare-associated infections are resistant to one or more antibiotic. And this problem is incredibly nonlinear because as patients are infected for longer, because it takes longer to find an antibiotic that works, both the bacteria from their infection can be transmitted to other patients, but also the genetic material that enables the resistance to antibiotics can be transmitted to other bacteria. So annually in the United States, this is causing 23,000 deaths, 2 million infections, and $20 to $35 billion in direct healthcare costs overall. So why don't we just discover new antibiotics? I think if we look at the history of antibiotics over the 20th century, it illustrates the point. We first discovered and deployed antibiotics in the 1940s. If we look at when the last novel class of antibiotics was discovered to treat gram-negative bacterial infections, it's only 20 years after that, in the 1960s. And then if we look at gram-positive bacteria, it's only 20 years after that where we have the last novel class discovered. And this isn't from a lack of trying. From the publicly disclosed discovery campaigns from companies like AstraZeneca or GSK, they together ran more than 150 discovery campaigns over a decade and turned up 0 useful leads that they could progress further. And if we look jointly across the industry in the development pipeline to treat gram-negative infections, there's only 2 novel antibiotics actually in development and less than 15 total. And considering the rate that things fail in clinical trials, this is pretty scary. So we think that combinations of drugs could be a rich source of new antibiotic material. And I think I'm going to tell you about 2 strategies that we think for why we believe this, and a technology that we're developing to enable this. So the first strategy is to recognize that gram-positive bacteria actually have a much larger set of antibiotics which can be used to treat them. This is because gram-negative bacteria have an impermeable outer membrane that blocks the entry of a large fraction of our arsenal of antibiotics. But serendipitously, we found, not our group, but others in the field have found that you can combine drugs which are known to be safe in humans, such as Imodium, which is normally used to treat diarrhea, and can actually disrupt this outer membrane and potentiate the uptake of antibiotics into the cell, therefore allowing us to broaden their usage against new pathogens. The second strategy is to recognize that our current drug development paradigm is poorly suited for this kind of program. Normally, we target our drugs against a single protein, but it turns out that very few proteins, if you were to remove them entirely, would actually kill the bacteria. And so this is because the proteins actually buffer each other's function. And you might need to block 2 at once in order to actually kill the bacteria. And this could be achieved by combining 2 drugs that have these both independent functions. So the way that we've discovered almost all clinically deployed antibiotics is actually just by taking the bacteria, combining it with the drug, and seeing if it stops their growth. And right now, we can operate at about a million scale tests. So we could test about 1 million drugs independently, or 1 million independent tests. And this is an issue if we want to actually test combinations of drugs because of this combinatorial explosion. So you can see in the first strategy where if we were to take the 2,000 or so FDA-approved drugs and cross them against 50 or so antibiotics we normally use to treat gram-positive infections to test their activity against gram-negatives, this would lead to 1 million tests, which is roughly at the scale that you'll see at a highly industrialized, high-throughput screening setting. And if you were to test in the second strategy, all pairs from 2,000 drugs, that would already lead to something that's tenfold larger than our current capacity. And that's ignoring the fact that you would be doing this against different pathogens, different resistance mechanisms, et cetera. So we need a new paradigm for screening combinations of drugs. The current paradigm is to use these very complex robotics facilities that are difficult to scale. And we want to replace them with this tiny rubber chip, which is about the same size as a 96-well plate, if you're familiar with that medium. And we can do this at a tenth the cost with maintaining the same throughput and also with minimal capital equipment. Right now, this device is totally hand-operated, and that gives us the power of industrial screening right on our benchtop. And we're also looking to further automate this to even improve throughput beyond the state of the art. So how does our platform work? You can imagine you take your antibiotics and your drugs, and you can emulsify them into tiny water droplets. And the droplets are so small that it's easy to manipulate millions of them at a time. We can create the combinations of drugs by just combining in pairs, random pairs of antibiotics and drugs together in these droplets, and then seeing if bacteria are able to grow. And the way that we do that is this rubber chip actually has 100,000 little microwells. Each microwell holds precisely 2 water droplets. So you can imagine if you have all of these droplets, each carrying different drugs, pooled all together, you can then scatter them across these wells, and they can pair up spontaneously into these combinations. We can then merge the droplet to mix the drugs together and then watch for bacterial growth. What does this look like in practice? You can see on the left, we have lots of different colored droplets. The color of the droplet is what encodes what drug or antibiotic each one is carrying. So therefore, we can look at each one of these microwells and identify what pair of compounds is present. On the right panel, you can see bacteria, which are in white, against a black background. And you can see that there's many spots where bacteria haven't grown. So then we can look at those spots and identify what pair of drugs is present in each one of those wells that stop the bacteria from growing. Right now, we're currently operating at 40,000 combinations per day at about 10% the cost of the state of the art. And that's totally by hand and on our benchtop. With further automation and scale up, I think we can hit 1% costs and a 10x throughput gain against the industrial state of the art. And we're also developing applications in cancer and other disease areas. So please come to our poster if you'd like to talk about antibiotic resistance or combinations of drugs and drug repurposing. And I'd like to thank the Deshpande Center, including the entire Deshpande team, and of course, our catalysts, Lori and Christine. Thank you.